In this video, I will be walking you through the steps involved with doing target analysis for In-Cell Westerns, ELISAs, and cell analysis. So we're going to open up a project here, and I'm going to choose a experiment that has already been started. Now, with all experiments within Empiria, when you create a new analysis, you give it a name right here at the beginning, put in any kind of description that is meaningful for you, and then choose the images that you have acquired. So next you're going to set up a template, and so you can either select a template that you have already created or create a new one from here. This one is fairly straightforward. We just have a set of wells that are background, and then this is a time course of drug that was added to the cells, starting with the controls here, and then the longer duration of the drug that was added. And so then these are then considered to be replicates of each one of the samples, and you can see that these wells correspond with the different conditions right here. So from here, you place you can move the grid if need be to get it into the correct location so that it isn't enclosing all of the wells right here. You choose the normalization channel, and here this was used with the cell tag 700, so we're going to choose that for the normalization. This 5% trim values, if by chance you have any saturations within your wells, this can account for that, and so you can continue to use that well for analysis. You can also look at the analysis table at this point. So for the different channels, we have this little mark right here that is saying that this is the normalization channel. And then for the 800, which is our target, we can look at the various calculations on this table right here. You can also click this button to export the data out to an Excel file. But you can also do that at the end of the process. So here we have, these are the different time points for each one of the samples. Each sample we can click and choose which targets or which samples to include on this. And then these are all given the same name. And then here we are showing either the normalized signal or the fold change over the resting cells. We can look at this as a scatter plot or a bar chart. And so you can see that as longer duration with the drug, we did see that ERK phosphorylation did go up at 30 minutes and then started coming back down at 60 minutes. So then here on the final reports, we have all of the information that we entered earlier as far as the experimental conditions, the template that was used for this analysis. And as we scroll down through here, we see that each one of the channels that were included in the acquisition has the full set of data tables. And again, if you click on any of these individual tables, you can export them out to Excel. And as we scroll further down, we do get the summary along with the chart. And then at the very bottom, we have the observations, next steps, and then the summary. Now at this point, once you have completed everything, you can export this as a PDF report so that you can print it out or place it into an electronic lab notebook. You can also export this as an experiment file. So if you would like somebody else to evaluate your analysis, you can have them do that as well. Thank you.